Dear friends, all my teacher colleagues, and my dear students, myself Dr. P.K. Verma, you all are most welcome in P.K. Verma biology classes. In the last 8 or 9 sessions, I have discussed different aspects of cell biology, and lastly I had discussed cell cycle and mitosis. Today I am going to discuss meiosis or the meiotic division, popularly known as reduction division. Sometimes the students think that it is a very tough and very long topic, but it is long, no doubt. It is not too much tough. It is simple. You have just to take proper attention in the first quarter of the chapter. It is a very essential chapter because the entire reproduction mechanism and the science of genetics depends on the spore and gamete formation. The variations depend on recombinations and crossing over, and these all are because of meiosis division. Now come to highlight some of the historical facts. Meiosis is a highly specialized cell division found in the germ-line cells in sexually reproducing organisms. It means meiosis occurs only in those organisms in which sexual reproduction is found, and it occurs on the germ cells, germline cells that will produce a spore or gamete, but it does not occur in the somatic cell that makes the general body organization. In 1859, Hofmeister studied division in pollen mother cells, that is formation of the pollen grains, and reported presence of nuclear filaments. Later on, Van Beneden reported reduction in chromosome number during gametogenesis in Ascaris. This popular name meiosis was coined by Farmer in 1905. One of the colleagues was Murray, and so sometimes it is written Farmer and Murray. Meiosis is such a cell division in which after the interphase, two subsequent divisions of chromosomes and the cells take place, leading to formation of 4 haploid daughter cells. That is, in the daughter cells, the number of chromosomes reduces to half the number of chromosomes present in mother cells. Now, before cell division, there will be interphase. There will be G1, S and G2 phase. So, in S phase, DNA duplication will occur, forming duplication in the number of chromosomes. So, normally in the mitotic cell, 2n chromosomes form 4n chromatids. When they divide to form two cells, 2n plus 2n, the equal number of chromosomes are distributed. But here in case of meiosis, after a single interphase, two consecutive cell divisions will occur. Two cells from one cell and again two cells from each cell. So there will be total 4 cells, and in these 4 cells the chromosome number will reduce to half. Normally the cells have diploid number of chromosome 2n, so the half is haploid or monoploid. But if a cell is tetraploid 4n, its half will be 2n. If the cell is hexaploid 6n, the half will be 3n. So monoploid is the single set, but haploid means half set of mother cell. In the G2 phase of interphase, chromosome condensation is poor. It also takes shorter time, and the nuclear volume is much more greater than the mitotic cells. Therefore, it is said that in the mitotic cell N/C ratio is lower, and in meiotic cell N/C ratio is higher. The cause of larger size of nucleus is because a number of events would take place inside the nucleus. Meiosis is a very long and complicated process of division that comprises two series of divisions marked meiosis 1 and meiosis 2. Each division will be followed by a cytokinesis. In both parts, meiosis 1 and meiosis 2, there will be prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. So the names are common: prophase, metaphase, anaphase, and telophase. Now come to meiosis 1. It is known as heterotypic division. This name is because at the end of meiosis 1, when two daughter cells will be formed, they will have only one allele of any gene. Normally it is said that a gene has two alleles. One belongs to mother and another belongs to father. But the two cells after meiosis 1 will have either one of the allele, either of the mother or of the father. So the two cells are different genetically. In prophase 1, nuclear volume is very high as compared to prophase of mitosis, and the events inside nucleus are identified as leptotene or leptonema, zygotene or zygonema, pachytene or pachinema, diplotene or diplonema, and finally dikinesis. These are different stages within prophase 1, and the chromosomal behavior changes markedly in each of the phases. In this table, all the stages have been written. The first column shows stages of meiosis 1, namely prophase 1, comprising leptotin, zygotin, pakitin, diplotin, dikinesis. Metaphase 1, anaphase 1, telophase 1, followed by first cytokinesis. These two daughter cells enter into interkinesis stage that may be a shorter or longer stage. It is different from interphase of cell cycle. Here you can see G1 phase and G2 phase, but there will be no S phase or DNA synthesis phase. After a short or long pause, the two daughter cells will enter into meiosis 2 and in meiosis 2 also there will be four major stages: prophase 2, metaphase 2, anaphase 2, telophase 2, followed by second cytokinesis. Therefore, four haploid cells are formed. Two of them are of one type and another two are similar but different type. In leptotin or leptonema stage, the chromosomes appear as very thin threads within nucleus. At first these are very long and form interovan network, but due to condensation they gradually become shorter and thicker and separate. They start roaming within nucleoplasm. In zygotin or zygonema stage, the homologous chromosomes come close to each other. They arrange themselves parallel point to point. That is, A gene faces A, B gene faces B, C gene faces C. This is point to point pairing, known as homologous pairing. Now a large number of contractile protein fibers develop between the histone coats of homologous chromosomes and pull them as close as possible. These protein fibers form synaptonemal complex, and such type of homologous pairing is known as synapsis. Because they become very close laterally, they look one double thick thread. In pakitin or pakinema stage, the common protein coats of each chromosome dissolves, and therefore two chromatids of each chromosome gradually separate, forming tetrad of chromatids in each pair. However, the true chromatids of every chromosome remain attached at centromere. These two chromatids of a chromosome are sister for each other, but the chromatids of homologous chromosome is called non-sister chromatid. It is very interesting that in pakitin stage, at some specific point known as recons, the non-sister chromatids break, make exchange, they refuse and form crossing over. Under microscope you can see crossing these chromatids each other. They become fused, forming the chiasma. These chiasma become clear when the chromosomes separate from one another in diplotin or diplonema stage. In longer chromosome, more than one chiasmata are seen, but in shorter chromosome, only a few chiasmata can be seen. Therefore, it is said that chiasmata frequency is proportional to the length of chromosome. There are only a few cases of meiosis in which there is no crossing over, no chiasmata formation, such as in case of male Drosophila. Otherwise, it is a common event. It should not be called an accidental phenomena. You will study crossing over in details in your genetics classes. In this figure, you can see that there are two chromatids in a homologous pair. One is painted blue, another is painted red. The two chromatids of blue are sister for each other and two red are sister for each other, but the red and blue are non-sister. Now, towards one end there is breakage and refusion in between two chromatids. Because of that, some part of the maternal and fraternal chromatids have become exchanged. It helps in mixing of character known as recombination. In this photograph you can see that the chromatid of one chromosome fuses and makes exchange with the chromatid of another chromosome, so a cross configuration or chiasma is formed. Here one chiasma is here and another is here. There were more chiasmata, but in the photograph the length has been decreased to show the clear exchange of the chromatids. Now synaptonymal complex breaks and dissolves. The chromosomes in every pair repel each other, as a result of which the chiasmata slip away towards two ends of the chromosome. That is known as terminalization of chiasmata. Once all the chiasmata are lost, diplotin stage is over and dikinesis begins. So in dikinesis, two chromosomes are completely separate from each other. There is no chiasma, but they remain together. There is no attachment of the two chromosomes, still they remain together, remain in pairs. By this time the centrosomes divide into two centrioles and start moving away towards two poles. Gradually the nuclear membrane dissolves and prophase 1 is over. Now in metaphase, all the pairs of chromosome move to equator and form the metaphase plate. Here the homologous pairs are maintained. So one of the chromosome is towards one pole and another chromosome is towards another pole. Now the spindle fibers develop and bind the centromeres of chromosome of their own side. That is, the fibers from one pole bind centromeres of chromosomes of that side, and the another chromosomes become bound to the other pole. That means no centrosome, no centriole can bind both chromosomes of a pair. In metaphase 1, centromeric DNA does not divide. There is no opposing force from the poles, and therefore the two chromosomes of every pair move to opposite pole along with their two chromatids. That means one set of chromosome moves to one pole and another set moves to another pole, but it is not known which maternal or fraternal chromosome will move to which pole. There are chances, 2 to the power n types of chances, by which chromosomes can be sorted out. So, some chromosomes belong to mother and some chromosomes belong to father. In telophase, the two groups of chromosomes reach their poles and new nuclear membranes are formed around them. It is followed by first cytokinesis. In this figure, you can see 3 pairs of chromosomes in prophase 1. There are chiasmata, obviously. These are in diplotin stage. In metaphase 1, these are arranged along the equator in the middle line of the cell. In anaphase, out of 3 pairs, 3 moves to one pole and 3 moves to another pole. So, 2 cells are formed that have some chromosome of mother and other chromosomes of father. The cytokinesis occurs in normal way by constriction in the animal cells and by cell plate formation in the plant cells. These 2 daughter cells have different sets of genetic characters because only one type of chromosome made of identical chromatids are present in them. So only one type of the allele of every gene is present. Meiosis 1 is commonly called reduction division. It is a 100 years old concept when scientists thought that the chromosomes are made of 2 chromatids. If it is reduction division, that is from 2n, the daughters have only 1n, then there is no need of second division. But in fact, the 2 chromatids are 2 identical chromosomes that will behave as a complete chromosome in future. So technically these are diploid. These are 2n. And after second meiosis, from these diploid cells, 2 haploid cells will be formed. So without meiosis 2, no actual reduction in chromosome number occurs. If meiosis 1 or meiosis 2 fails, the daughter cells are diploid. Meiosis 2 is called homotypic division because from each cell, 2 similar cells are formed. It is a process very similar to mitosis in the haploid cells, in which after interphase each chromosome is made of 2 chromatids, and here also after interkinesis the chromosomes are made of 2 chromatids. From each daughter cell of meiosis 1, two identical cells are formed in this stage. So it is homotypic division or equational division. It has 4 stages: prophase 2, in which chromosome recondense if there is a gap, if there is a considerable length of interkinases. But in many cases, meiosis 1 immediately begins. Meiosis 2 immediately begins after meiosis 1, and there is no interkinases stage. So if there is interkinases, if the two cells separate and meiosis 2 begins after a few hours or a few days or a few months, in that case the decondensed chromosome again become condensed, recondensed. Chromosomes with both the chromatids attached at centromere become visible. The nuclear membrane dissolves gradually and the centrosome redivide to form two centrioles. These centrioles move at right angle to the earlier axis. That is, if there was a vertical axis in meiosis 1, there will be a horizontal axis in meiosis 2. So the centrioles move 90° left or 90° right, and the total angle is 180°. The direction of chromosome also moves to 90°. That is, they form equator at right angle to previous equator. That is parallel to the equatorial plane of earlier division. Spindle fibers are formed, and at right angle to them the equatorial plane, metaphase plate, will be formed. The unpaired chromosomes move around in the protoplasm and come on to the new equator formed at right angle to the previous equator. Spindle fibers from both the poles. Here chromosome has no pair. These all are unpaired. So the spindle fibers from both the poles join their centromeres from the two sides, and centromeric DNA divides at the end of metaphase 2, lastly in metaphase 2. So the two chromatids become completely separate after metaphase 2. In anaphase 2, these chromatids move to opposite pole, and therefore in each cell of previous division, two new nuclei will be formed in the telophase stage. In most cases, first cytokinesis forms two cells, but sometimes first cytokinesis is absent. So in a single cell there are 4 nuclei, and in second cytokinesis cell divides at 2 axes, forming group of 4 cells or tetrad of haploid cells, such as seen at the time of microspore formation in the flowering plants, mostly in the monocotyledonous plants. The cells divide normally by constriction or by cell plate formation to form 2 haploid outer cells. From the 2 outer cells of meiosis 1, 4 haploid cells are formed. 2 are of one type and another are of other type. Now these four cells shown here in the figure, these are the two cells of meiosis 1. Say it is A and another is B. So from A, two daughter cells of A type, and from B, two daughter cells of B type are formed after meiosis 2. Now at the level of DNA, these things have been shown here by the Polyster et al. In 1951. In normal diploid cell, chromosomes are 2n and the DNA content 2C value is 3.28 plus minus 0.07 picogram. After S phase, the DNA content increases to 4C minus delta and the measurement was about 6.28 plus minus 0.07. After meiosis 1, two types of cells containing N1 and N2 chromosomes have the DNA content again near diploid 3.35 plus minus 0.04 picogram. Meiosis 2 reduces the chromosome amount or the number of chromatid or chromosome again to half. So after meiosis 2, two cells of N1 type and two cells of N2 types are formed, and in each cell 1.68 plus minus 0.2 picogram DNA is present. This data has been given in the common textbook of cell biology written by D. Roberts and D. Roberts. Now what is the benefit or significance of meiosis? Meiosis is a process of division by which haploid gametes are formed in animal cells. In plants, haploid spores are formed. The microspore and megaspores in the flowering plants are formed by meiosis. In most cases, meiosis occurs before gamete formation, that is pre-gametic meiosis. It may be sporic meiosis. After fertilization, the zygote contains diploid number of chromosomes. So, if the organism is haploid, then meiosis will occur after fertilization, that is zygotic meiosis, such as in the algae bryophytes. The organisms are haploid, so meiosis occurs in the zygote. But in higher plants and animals, meiosis occurs before gamete formation. In diploid organism, it occurs to form haploid spores, that is sporic meiosis, such as in the pteridophyta, or gamete formation, gametic meiosis, such as in case of animals. These are pre-fertilization meiosis. In either way, haploid-diploid relationship is maintained, and in every species a normal and consistent number of chromosomes is maintained. So meiosis will occur before fertilization or after fertilization. Meiosis yields 2 to the power n types of variations in spores or gametes, and when fertilization occurs, 2 to the power n into 2 to the power n, that is 2 to the power 2n types of genetic variations are formed in the offspring. Crossing over among the chromosomes further increases chance of variation. These variations help in adaptations and evolution of organisms. Now, please revise the topic, view the video twice or thrice, and make everything clear in mind. Only after that you can understand the life cycle of various plants, sexual reproduction in plants and animals, and the subject called genetics. See you in the next session. Thank you very much.